Hello, my name is Sam Boland, and I'm a second-year PhD student in the Department of Biomedical Engineering. And I researched the link between traumatic brain injury and Alzheimer's disease, which we believe may lie in our vascular system. First and foremost, I work in a mechanobiology lab, which means that I study how cells sense and respond to mechanical stimuli. The tissues in our body are composed of cells and a fibrous network called the extracellular matrix. During an injury, the extracellular matrix is deformed, and this applies a force to cells. And that force is transmitted to the cell nucleus through rigid cytoskeletal components and a series of signaling proteins. And this can affect things like gene activation. So essentially, when we apply forces to cells, we have the potential to change things like what kind and how much protein a cell produces. Mechanobiology plays an essential role in the healthy functionality of cells and tissues, but extreme forces can have extreme impacts on cellular function. Each year, over 1 million Americans experience traumatic brain injury, but I'm particularly interested in studying individuals who experience multiple mild head traumas over the course of their career. This might include athletes such as football players or active duty members of our military who experience concussive blasts when they are exposed to explosions. During these impacts, force is transmitted to neural tissue and ultimately to the vast network of blood vessels that supply oxygen and other essential nutrients to our brain. And we believe that this force transmission can cause changes in gene activation and overall functionality in the cells that compose our blood vessels in the brain. This is important because we recently learned that healthy blood vessels play an important role in preventing Alzheimer's disease. Alzheimer's is caused in part by the aggregation of a protein called amyloid beta, which is a waste product produced by neurons. Healthy vascular cells express a protein called LRP1, which binds amyloid beta and transports it into the lumen of blood vessels where it can then be cleared from the body. But when vascular cells stop expressing LRP1, they can no longer clear amyloid beta, and it begins to build up in the brain, ultimately contributing to Alzheimer's disease. We believe that the forces experienced during traumatic brain injury may alter vascular cells' expression of LRP1. For my thesis, I'm designing an in vitro model of vascular trauma to help test this hypothesis. I'm using high-speed linear actuators to simulate traumatic brain injury in vascular cells by rapidly stretching them. Then I use a high-powered microscope and genomics techniques to assess the functionality of vascular cells, particularly looking at the ability of vascular cells to modulate blood vessel diameter and clear proteins like amyloid beta. I hope that this research will help improve our understanding of the pathology of Alzheimer's disease and may one day lead to improved treatments. Thank you.